All right. Well, good morning, good afternoon, everyone, and welcome to today's Exploring by the Seat of Your Pants event. My name is Joe Gorewski, and I'll be your host for today. What can I say about today? Today's a busy day. We have 5 live events coming your way. Just this morning, we were exploring Everest with Alex Hars, who put together the Everest Quest expedition and an incredible VR film. We were able to share that film with students in advance. They were able to meet with Alex today. He was able to answer questions, take them behind the scenes. It was pretty cool. Later today, we'll be with Alex again for a second session. Then we've got a session with our friend Sonia, who's going to take us into the mesophotic zone of the ocean to look at the biodiversity there. It's not discovered there very much, and there's tons of new species to be found. And right now, we are going to head to Zambia for a live event from the field, which, as you know, is pretty much our favorite live events to do here at Exploring by the Seat of Your Pants. Last week, we joined Joe Cutler. Joe is an ichthyologist, freshwater ecologist, and an African river explorer. He's also a National Geographic explorer. He joined us at the start of a new expedition at the Kabompo River in Zambia, and they were just getting ready to start. They were down in the gorge. They were camping by the shore. We got to talk to Joe about his last expedition in Angola and the work that he was doing there. And then the last week, they have been out in the field and making their way down the river. They've been fishing, collecting samples, and we're going to connect with them live right now. It's kind of the end of their day there in Zambia. I think it's around 5 o'clock, so they're probably thinking about some dinner and wrapping up the research. So let's grab Joe from backstage. Let's go live in Zambia. Here we go. Hey, Joe.
Hey, Joe, how you doing?
Good, Joe. It's so good to see you. I think your hair and beard have grown even more since we saw you last.
Yeah, every day they get longer.
Yeah, very cool. Well, Joe, it's great to have you. We've obviously got lots of stories to catch up with. I've been seeing little pictures and video clips from the field. We're really excited to be spending this time with you, the team, and seeing what you've been up to.
Yeah, I'm really excited to share our updates. It's been a great adventure thus far, and everybody's in good health and good spirits. So that's most important, but it's been a beautiful trip down the Kabompo River, and we've learned a lot and discovered a lot already. So I'm really excited to share.
All right, excellent. Well, Joe, give us like maybe a minute or two and tell us what the week's been like so far, and then maybe we can share a few images before we explore a little bit.
Yeah, so the week started off pretty hectic, actually. So where we launched our boats, there were a ton of big rapids, and the first day on the water was wild. I mean, we were paddling down waterfalls that were 10 feet high. We were running rapids, and the rapids that were too big, we had to portage around and carry our equipment and our boats. And there was a lot of roping boats down the rapids and slippery rocks and falls. So that was pretty hectic. On day 2 of the expedition, we actually paddled past the outflow from a large copper and nickel mine. So an area where I expected really heavy impacts, but actually the river turned out to be quite healthy. It seems like that mine is treating their effluents quite well. And then we've now entered into West Lunga National Park, and we're starting to see some wildlife. So we've seen impala and crocodiles and even a hippo today. So that's quite exciting. And of course, lots of good fish. So that's all been very interesting. And yesterday we had a bit of a scientific mystery, which is always exciting. Joe, I don't know if you want me to dive into the scientific mystery immediately or wait a little bit and then I can kind of dig into that one.
All right, Joe. Well, I think you've brought it up. We don't want to keep them in suspense. So let's dive into that mystery and then we'll share a few images.
Great. Well, yeah. So we're paddling into the game management area just outside of the national park, and the ranger who we were with alerted us that there was a hot spring that would flow into the river. So, everybody's all excited about a hot spring. So we decided to paddle up and check it out. And immediately you could feel that the water was warm. It was crystal clear. Whereas the Kabompo behind me is actually relatively green and brown. So we paddle up this nice warm hot spring, and I look into the water and the fish look weird. They've got these big buggy eyes and they're swimming head down, which is an indication to me that these fish might be sick. And so we spent a lot of time sampling that little hot spring, trying to figure out why the fish in the hot spring are sick and why the fish in the main channel are not sick. And I have a hypothesis. So my hypothesis is that during the big rainy season, when all of this floods, there's not much difference in temperature between the hot spring and the main river, which allows fish to go in and live in there. But as the water level drops, those fish become isolated by the change in temperature. And then they're stuck in this little freshwater lens. And in warm water and sulfuric water, fish tend to get disease. So my hypothesis is it's related to seasonal changes in water level and water temperatures between the stream and the hot spring. Now I'm not sure what disease it is. It could be fungal, it could be bacterial, it could be a virus. That's something that we're going to have to figure out in the lab when we get home. So always exciting to have a scientific mystery and something that I've never seen before, despite 15 years of field work in Africa.
All right, Joe. Very cool. I mean, there's anywhere you go and explore our planet, we're going to find things we don't understand yet. And you're doing exactly what a good scientist should do, forming a hypothesis. And then, maybe we'll get to the bottom of that mystery. Maybe it'll take a little bit longer. I guess we'll see. All right. I think we had a pause. We have to remember Joe's using a Starlink in Zambia. So we're just have a little bit of a pause here. Hopefully things will reconnect for us. Oh, there we go. I think we have Joe back. Joe, we got you back.
Got you back, Joe.
Perfect. All right. Well, you talked about some hairy conditions on the river. So I want to open up some photos here, and we're going to take a look at some of those conditions, what this river looked like. So this should come up now. And we can see from your last expedition in Angola, you're using a different mode of transportation. You're not using those replica dugout canoes.
Yeah, well, our first 2 days we were using those inflatable boats, and they're much better suited for whitewater, as you can see there. We've actually switched now to a different type of canoe. If I lean back, you should.
Yeah.
We're past whitewater, so that's good. And others.
All right. We lost you for a sec, Joe, but we had a good look at those more like your traditional canoes, and you're past the whitewater now. So that's good.
Yeah, we're past the whitewater for now. I think we've got a few more rapids ahead, but those we should be able to portage in the canoes.
Yeah. So a few more photos here going down some of those waterfalls you were talking about.
Yeah, we're really lucky on this expedition to have a great storyteller with us. Eric is a phenomenal videographer and photographer, so he always runs the rapids first, and then he's there with his camera ready to capture shots like this as we navigate our way down the river. And this was one of the big ones. We were getting wet up to our shoulders coming down this rapid here.
All right. And then sometimes it looks like they were probably a little too strong.
Yeah, the river dropped a lot of elevation through that stretch. So we were better off actually pushing the boats around the rocks, through the trees, getting them down safely, which is much better than swimming in those rapids, because sometimes there'll be a crocodile sitting at the bottom of the rapids waiting for an easy meal. So always a bit treacherous around the whitewater.
All right. And then, of course, sampling every day.
Yeah, we're doing a ton of science on this expedition, and all my expeditions have a big fish bent. So this is a shot of us electrofishing, and it's a really cool way to catch fish. And we have been catching a lot of very. All right, got you back.
All
I
right.
was just saying, Joe, when we're done going through the photos, I've actually got some some live fish to show off just now.
Yeah, we'll be there shortly. So a little hygiene on the river, maybe cooling off in this
Yeah,
photo.
actually, yeah, you know what's funny? I actually took my first bath of the expedition today. So, I've rinsed my face a few times, but today I actually got out a bar of soap and washed my clothing and my body. I always prioritize research and fish sampling over getting clean. So that usually falls to the bottom of the list of my to-do list.
All right. Well, nice of you to get nice and cleaned up for us today, Joe. Campsites. I mean, can't beat these views waking up like this.
Yeah, I always try to make my tent as close to the water as possible. I want to be so close that if I step out my front door, I could fall into the river. It allows me to listen to the catfish feeding at night, hear the frogs, hear the birds. I just love the sounds of the river. And it's such a treat to be able to sleep waterfront every night.
All right. This I thought was a cool picture. This is a cool looking spider.
Yeah, it's not just the big things that fascinate me. I love the invertebrates. So every now and again, you get the chance to really crawl around in the trees and crawl around in the grasses and see incredible things like this orb weaver spider. Um, I also am a big fan of the mantises. So every time I see a
Yeah.
mantis, I'll grab it and I'll put it on my hat.
All right. You were just telling us how you grab the mantis and put them on your hat. I saw one of those pictures from Angola uh, in the WhatsApp
Yeah,
stream.
it's always nice to have a friend on the expedition. And I really like the mantises because they eat mosquitoes. So the more mantis that are around, the fewer mosquito bites you get.
All right. That sounds pretty good. And then I like this shot too, kind of a action shot hiking as the sun's going down. Fishnet ready to go.
Yeah, this was us headed out to the source Kabompo River. So, the source area is so equipment. We brought everything that we needed to do the research. So our nets, our drones, our water quality probes, two line for this river.
Okay. And then our aquariums. I think what I like the most, well, I mean, I like a lot of things about the expedition, but I love that you brought this little photo aquarium with you so that you can show people who live around the river what the biodiversity is like. All right. I think we'll have to be a little bit patient today. The Starlink's really acting
We're in
up,
the field. No, it's
but
all
that's
good.
okay. We're in the field. Uh, all right, we got you back, Joe. We are just talking about showing the biodiversity to those who live along the river and maybe haven't seen it before.
Ooh. Oh, it's back now.
Okay,
Uh,
perfect.
Great. Yeah, the photo aquarium is such a treat because, people here, everybody fishes. Everybody is used to the fish, but the local Zambians will probably have one name for a whole group of fish. So every minnow is just one thing. So when you get the photo aquarium out, you can really see the details and the distinctions between some of these fish. And I love it. We almost always, when we're sampling fish, we've got a crowd of people around us. And, I'll be learning the names in the local dialects, like in Lunda, but I'll also be sharing some of the common names and the scientific names and just kind of sharing my enthusiasm for the fish with anybody who's willing to listen.
All right. We're going to leave it there with some of those photos, Joe. And, I know you have a little bit of stuff to show us today, so I'm going to make you nice and full screen.
Great. Well, I wanted to show you all a bit of my sampling kit for fishes. So I've brought some of it down and laid it out on the beach. So let me see. Right here, I've got my fish trap. So I put this out every single night at every site. Fish will swim inside of it during the night, and then in the morning we get to check them out. I have right here, I've got what's called a fyke net. And it's kind of a combination of like a long wall and then a series of chambers. And the fish will swim into those chambers and also get stuck, and then we can sample them in the morning. I've got my handy dandy cast net here. And this is one of my favorite ways to sample. It's got a bunch of weights at the bottom, and I can use it to throw it out in the water into an open space like this, and I can catch a bunch of fish. And then my other major technique of fishing is with a dip net. So in shallow water or on the shallow banks, I can use this to catch small fish. And a lot of the diversity are small fish. So I've got my little photo aquarium here right now, and you can see this would be a typical catch. And when you really look at them, they all look pretty similar, but actually you'll start to notice some distinctive characteristics. And it might surprise you when I say that there are actually 3 species of minnow in this photo aquarium right now. And they're distinguished based on their color patterns, their body shape, and their size. So I've got 3 species of minnow all caught in the same net. I scooped them all at one moment, and here we go. This is a smattering of the diversity in the Kabompo River. Now, I know that not everyone is excited about small fish, so I've got a big fish to show off here. I can't claim that I caught it. One of my colleagues caught it. But let me grab this fish here. Oh, this is a tigerfish. And it's
Oh
named a
yeah.
tigerfish because of those teeth. And it's an impressive predator. So this is the top predator in the Kabompo River system other than the crocodiles. And so we've got this fish. It's probably, you, 5 or 6 kg fish. So, a 10 or 12 pound fish. And it's going to be a feast for dinner tonight, which is a real treat after eating rice and beans for the last week. So those are a few of my show and tell moments. Uh, with the excitement around that fish, I've got a few team members here hoping for one more. And we've got a crowd of rangers around all excited to have a big meal tonight. So that's one of the great things out on the river. You never know what you're going to discover or run into or encounter, and it's really wonderful to share experiences and to share a good meal with friends and with strangers alike. So that's a bit of my fish sampling kit, and we've already caught 14 species of fish at this site today. So lots of exciting science to do with those fish.
All right. Joe, that's really cool. I know you were last week, you were talking about tigerfish and how you weren't sure if you'd encounter some. So that is a beautiful fish. Those teeth are pretty impressive. We're not used to seeing teeth like that on a fish. Oh, I think we might have lost Joe for a second. We'll just wait a second. Like I said, Zambia, Starlink, pretty remote in the field. So the fact that the connection is this good, so clear, we can see Joe, the river, those fish that he's been catching is pretty impressive. While we are paused and we wait to see if, yeah, here we go, refresh happening.
Oh, I got you back, Joe.
Perfect. I was just saying, Joe, I'm so excited we got to see that big tigerfish. Those are impressive teeth.
They are impressive teeth. You wouldn't want to mistakenly, put your hand in its mouth. And, I always wonder if you're swimming around with some jewelry on your ankle or something, maybe they might get a little bit confused and come give you a nip. But,
Yeah.
we've also heard that there are really big crocodiles living here. So there's plenty to be afraid of in the water. And usually by the time the sun sets, we are far away from the river. And, as I said earlier, we saw a hippopotamus today, and it was running away from us, thankfully, rather than running towards us. So I'm sure we'll run into a few more as we go downstream.
All right. I forgot, Joe. I have one more clip here, a little river clip where you dipped your camera into the water. So I think that'll be
Great.
fun to share before we go to Q&A action. So
Perfect.
yeah, let's see what it looks like beneath the surface. There we go. Now, Joe, is this a clip from in the spring?
Yeah, this is a clip from inside of the hot spring, and you can actually see some of those fish have big bulgy eyes. You can see
Yeah.
that the water is crystal clear but tinted blue, and that's because of, the chemicals in the water from the hot spring. And one of the things that's really interesting about that stream is not every fish is diseased. So some of them are diseased and others are not. And we're really curious if there's not some sort of adaptation to living in those conditions and disease resistance in certain lineages of fishes. So that's a question we are definitely going to be digging into when we get these specimens back to the lab.
All right. Well, Joe, we're going to get to some Q&A action now. Those who are tuning in via YouTube, don't be shy. Use the chat sidebar to send us in some questions. So we'll start. We've got a few questions here from Miss Coburn's crew in Hamilton, Ontario. First thing they want to know is when you're going down to the river, when you're sampling, when you set up your camp for the night, do you often have a lot of local people come to visit? And have any of them ever joined your team?
We often have a lot of people come to visit. Actually, right now, we've got a whole crowd of people with us. Um, and it depends on where you are. So sometimes you're in the middle of a really remote area, like right now, we're inside of a national park. The crowd who's come to join us tonight are other park rangers. They just came down because this is their fishing spot, and then they ran into us, and now we're going to share a nice meal. Um, but anytime we're staying near a community, we are a major curiosity, and we will have crowds of people around our camp, around our science, and around our kitchen. So I really enjoy kind of sharing the experience and especially sharing the science with local community members. And yes, we do have community members join us. We've got a ranger from West Lunga National Park right now. We've got several Zambians on our team who live beside the river and, were fishermen before they were working with our project. And sometimes, there are some scenarios where, you need to change your team composition halfway through an expedition and you need another expert on the river. And thankfully, in these riverside communities, people know the river. They know how to paddle. They know how to live off the land. And so it's actually really wonderful. It allows us to learn a lot from the local communities as we go downstream. It's one of the great parts of this work, is learning from local community members and local experts and involving them in the work that we do.
All right. Another question here, Joe. We know how you showed us some technology, some gear you use to catch the fish. We know you're obviously doing this as a way of outreach, but how else might you share your findings with the public?
Well, the ultimate goal is publishing in peer-reviewed journals. That is the epitome of science. But oftentimes that takes a long time. So we have two approaches. We have, kind of the social media approach, which is really sharing with the public. So I work for the Wilderness Project, and we've got an Instagram page, and we are posting on that. If you check our
All right. We have another little pause here, but in the meantime, I'm going to grab the Wilderness Project and I'm going to share their YouTube page, or not YouTube, their Instagram page. So if you want to check it out, you should be able to find it. There we go. And I'll drop that into one of our banners. So if you want to check out that Instagram page, you can do that there. Wilderness Project. And we'll give a Starlink a second to reconnect and go from there. So again, I've got some great questions coming in via the chat. I can see we have a good crew tuning in. So don't be shy. If you have some questions for Joe, don't be shy to send them in. Looks like Joe had to exit and come back in. So we'll take away the pause, Joe, and we'll bring in the live, Joe. There we go. We got you back, Joe.
All right, I'm back on now.
Yeah.
And I wanted to correct myself. Our Instagram page is the Great Spine of Africa rather than the Wilderness Project. So if you're looking to check us out at the Great Spine of Africa.
All right, we'll put that banner up there. Great Spine of Africa. There we go. All right, Joe, we got that up there for people to check out. Very cool.
Perfect. Thanks,
another
Joe.
question.
And then,
oh, sorry, go ahead, Joe.
Go ahead. Oh, I was just gonna say the other way that we share our data and is that we write reports on every expedition, and so those are on our website. And again, they take a little while to put together, but then they get put online so anyone can follow up and see what we've been seeing. Obviously, it's not just fish; it's wildlife, it's human activity, it's infrastructure, it's kind of everything.
Okay. So I've got a question here. Obviously you collect some specimens to bring back, but sometimes you're just collecting and you're going to release them. So what are you doing to try and, as you're looking at them, maybe doing some measurements to try and keep them alive before you release them?
So the main thing about keeping fish alive is fresh water and plenty of it. So the more fish you put into a bucket or into an aquarium, the more they're consuming the oxygen in that water and the quicker they're going to keel over. So, if I've got a lot of fish, I'll put them in several buckets. We've got aerators to keep those fish alive. And then when it is time to euthanize them, then we use a special chemical to put them to sleep nice and gently so that the specimen come out as nice as possible. And when we have a lot of one fish, we can always put them back in the river. We just want enough examples so that if we happen to have an undescribed species or something like that, that we can then describe it with the specimen we're collecting. But the only stuff that we actually take home are fish and invertebrates. So we're not taking samples of hippopotamus or crocodiles on these expeditions.
All right. We've got a group here who's doing some budgeting in their classroom right now. So they're wondering, what's the budget like for an expedition like this?
Whew. That's not my job, I should say. But it is quite expensive. We have a large team. We've got 10 people on the water. We've got 2 people in land cruisers following us down the river. We've got a lot of food, a lot of science equipment. Now, a lot of that gets reused expedition after expedition, but it is expensive, and we're lucky to have a lot of donors. It's a non-for-profit organization. We don't make any money from the work that we do, and we're supported exclusively by donor dollars. And that has been really important to be able to conduct the work that we're doing. But it is quite a budget to pull this kind of work off. Very few projects can do the work that we're doing and have the funding support that we've got. So we're really lucky, and I'm extra lucky because I don't have a fundraiser for the team.
All right. Yeah, you get to do the fun stuff out in the field.
Exactly.
Running waterfalls and catching fish.
Yeah, and dodging hippos.
Yeah, I know you mentioned that you heard some stories that there's some groups up there that maybe aren't might not be so friendly when you get down to them.
Yeah, we got really lucky. So we had a warning that there was a pod of hippos that had been hitting boats and biting people. And we knew exactly where they were going to be. We were talking to all the local fishermen, and they were saying, they're just downstream, they're just downstream, they're just downstream. And suddenly the fishermen started saying, they're just upstream, they're just upstream, they're just upstream. And we had passed the group of hippos without ever even noticing them. So we got lucky this time. It doesn't always happen like that. And on the Quito River, my last expedition, I got hit by a hippopotamus, and two other boats got hit by a hippopotamus. So it can get really hectic really fast.
All right. I've got someone who is curious if you ever use rotenone to collect.
We don't use rotenone. So rotenone is a fish poison, and you can put it in the stream, and all the fish kind of go to sleep, and then you can just scoop them up as you'd like. Now that was a very common practice in the 70s and 80s, and it really did, expand our understanding of fish diversity, especially in remote parts of Central and Southern Africa. But it's become a bit of a taboo, so we're using manual methods that allow us to release fish that we catch that we don't need to kill. So we don't use rotenone, but it is a very effective technique. And in certain habitats, it might be the only way to sample.
All right. So there's a class studying geography right now. And one thing they're looking at is the interactions and movements between species. So in the last, since this expedition's begun, have you seen any interesting interactions between species? I guess for movement, that's kind of that spring that you found is kind of interesting, where they kind of get caught there seasonally. Kind of could be a good example.
Totally. Well, I mean, I think fresh waters are a great system to study movement and isolation. So you can imagine a river system with a waterfall halfway through it.
Hmm.
Some fish will be able to get upstream of the waterfall, other fish will not be able to get upstream of the waterfall. So there are these barriers.
All right, we'll give Starlink a minute to sort itself out. Hey, Joe. You're
Got you
back.
back, Joe.
You were just talking about the
Yeah.
waterfalls are a good barrier.
Yes, and then in a big open system like this, a species like a tigerfish might migrate, tens or hundreds of kilometers up and down the stream. So it's a really interesting, freshwaters and rivers in particular are a really interesting system to study biogeography and the distribution of species in. And in terms of species interactions, one of my coolest interactions I've seen recently, I saw an African fish eagle chasing a Goliath heron and having a mid-air battle, just fighting in there. And these are two incredible and large them having that
All right, that sounds like a pretty epic battle. Maybe we'll get Joe back in a second. In the meantime, I'll just let classrooms know, hopefully we'll be able to do one more of these river connections next week before the school year officially ends. There we go. So yeah, those are two big birds, Goliath heron and an African fish eagle. Oh, I don't know if Joe has my audio yet. We've got you back, Joe. You're coming through nice and clear.
Got you now. All right, Joe. Thank you.
Yeah. No, I was just saying those are two pretty big birds. That would have been a cool interaction to see.
Totally. It was like a once in a lifetime thing to observe.
All right. Well, Joe, you're in camp now. You're looking forward to a tasty fish dinner, but the work doesn't end. Are you going to be doing some more fishing tonight?
We've got a lot more research to do tonight. So, I've got to put all that fishing equipment out into the river. We're going to be putting out two audio recorders. So one that will be recording bat calls, one that will be recording frogs, and that's one of our other ways of sampling biodiversity. And then I've also got to do all the backups of all the data that we've collected over the day. So it's going to be a busy evening of work. But certainly punctuated by a nice big meal and a good time around the campfire. So no complaints on my end. I can't believe that I get paid to do this kind of work.
Yeah. So this was another shot that I had pulled aside. This is some evening work. Looks like maybe you're sorting some fish, taking some pictures.
Yep, yep. Every single species that we collect at every site, we photograph and we take a tissue sample for DNA. And sometimes that takes us well into the night. And I suspect it may be the case also tonight. So very important in the field to have a good headlamp, a trusty light for those late night science tasks.
And so, Joe, you mentioned different collection methods like the recording for biodiversity. You just mentioned DNA. Do you ever take samples to bring back and look at eDNA in the rivers?
Yeah, yeah. So one of the main reasons we're taking those DNA samples is so that we can understand our environmental DNA samples. So every 50 to 75 kilometers, we're taking eDNA samples, and we're also doing it at every interesting site. So it's much more common, and we are basically measuring the DNA of the river. So everything that's living in the river, everything that's touching the river is shedding DNA all the time. And we can then filter the water and get a little tiny bit of that DNA and study it. But one of the difficulties here in Zambia is we don't know who the DNA belongs to. So we've got to actually physically catch the fish, take a tissue sample, verify the ID, and then we can sequence that fish, and then that barcode is what we need to understand our environment.
All right. So for those who might not know, environmental DNA is what eDNA stands for. And it can be used in so many different applications like water, ocean, freshwater. We're using it more on land, soil, even the air. There's scientists working on, can we grab samples of what's in the air?
All
Looks
right,
like
Joe. It looks like
Starlink is
I'm getting you
battling
back.
back. We gotcha. All right. Joe, can you hear me now? No, sound hasn't come back yet.
Gotcha now, Joe.
Yes. All right. Well, I was just going to say, Joe, that's awesome. You're doing that baseline work, which is going to be really important for science, other biologists following in your paddle strokes in the future on the river.
Yeah, so many of these that we, and so it's really important to know what's going on today, what's the diversity, what are people doing, so that 5 years from now, 10 years from now, 100 years from now, we can compare what we've seen to what the next generation of scientists is seeing. So anybody tuning in, if you want to be an African, know that there's a lot more to discover. And
All right. We're going to let Joe go shortly so he can get the traps into the water. He can enjoy a nice dinner. Hopefully, we'll get one more little stretch of connectivity where we can thank Joe and let him continue with his work for today. Here we go. Starlink. There we go.
thanks
All
for your
right.
patience, Joe.
No, Joe, please. We deal with this all the time when we connect from the field. And the connection, in between is almost crystal clear. So being able to do this from the field instead of cell phone like we've done in the past is a treat to be able to see the details even more finely. I was just saying, we're going to let you get to your dinner, get to putting those fish traps into the river. Maybe if we're lucky, there's still one more week in the school year. Maybe we'll get to do this one more time. But I can't thank you enough, Joe. We love doing the live stuff from the field, and what you're doing is important work, and it's pretty fun.
Well, it's great to be able to share it with the next generation of scientists. So, Joe, thanks so much for including me. I really always enjoy these exploring by the seat of your pants hats, and love streaming into the classrooms. I hope that there's a future African freshwater ichthyologist streaming in right now, and I'll find my replacement through one of these calls.
You never know, Joe. You might have I think we lost him. right. We're going to say goodbye to Joe in just a second. A huge shout out to those who tuned in today, those who will be tuning into the recording. Joe, I think we got you back. I was just saying, you never know. There could be one of your future PhD students hanging out here right now. And they might join you in the field in 10, 15 years or so. Oh, I don't know if Joe caught that. All right. We got sound back, Joe? Sound? Can you hear me, Joe? Nope, not yet. Come on, Starlink. Give us a couple more seconds. How about now, Joe? Can you hear me now? All right. Funny thing with connectivity sometimes. We see Joe very well right now. I can hear the birds chirping in the background, but the connection for him being able to see or hear me isn't there yet. I think it's there now.
Got it.
Yeah.
Got it.
I was just saying, Joe, before we say goodbye, there could be a future PhD student of yours there tuning in right now. 10 years or so down the line, they could be out on the river with you.
I look forward to it.
All right. Well, Joe, thanks so much for connecting today. And, thanks to your team. Obviously, they're doing hard work behind the scenes and helping you make those collections and get where you need to get. So a shout out to them as well. And who knows, if we're lucky, maybe we'll have one more of these next week.
I hope so as well. Thanks so much.
All right. Thanks, Joe. Thanks, classrooms. We are going to sign off for now. Thanks, everyone.